Thank you, everybody, for joining us today. My name is Matt Hewitt. I'll be the moderator today for the panel, which is from concept to commercialization and living therapeutics. Hindsight is 2020, but how do we improve our vision going forward? I have today a very good slate of panelists, and I'm hoping to ask some questions which will stimulate some good discussion. But first, we're going to start with some introductions, and we'll probably start with Doug, if you want to introduce yourself.
Well, thanks, Matt, and I'd like to thank Arm for arranging this presentation. My name is Doug Dorfler. I'm the founder and CEO of Maxite. We're located in Gaithersburg, Maryland. We're a cell engineering company. We've developed a non-viral delivery technology that we've been developing for the last 20 years, and we currently have 14 commercial partners who use our technology, including companies like Sana and Allogene and Editas and others, which I'm sure we'll touch on later today. And I've been involved in cell therapy for the last about 25 years and really happy to be a part of this panel. So thank you.
Howard, would you like to go next?
Yeah, well, thanks, Matt, for hosting this. And it's a pleasure to be here and appreciate Arm's invitation. I'm the CEO of Brooklyn Immunotherapeutics. We are a platform company pursuing both cytokine therapies as well as mRNA engineered cellular products, including those that are intended for in vivo gene editing.
Great. Thank you for being here, Howard. Kristen?
Thanks, Matt. It's great to be here with you and my fellow panelists. I'm Kristen Urema. I am the Chief Commercial Officer of Atara Biotherapeutics. We are trailblazing in T-cell immunotherapy, specifically allogeneic T-cell therapy. And we're using our novel allogeneic Epstein-Barr virus, or EBV T-cell platform to develop off-the-shelf therapies for patients with cancer and other serious autoimmune diseases, including, for example, multiple sclerosis. We're using T cells from donors with healthy immune function to create products that are designed to reinforce a patient's own immune system and effectively tackle disease. And we're trying to really build upon the great benefits that, for example, autologous T cell immunotherapies have made available today, and also address some of their limitations. We're very excited with our manufacturing progress and our ability to store our finished product in inventory for quick, reliable, and easy access to patients. And we are the most advanced technology platform in the clinical setting among allogeneic T-cell therapy companies. We're in phase 3 and preparing for filings with the lead product, which is called Tab-cel, for a rare form of lymphoma transplant patients get. And we're also in a randomized phase 2 study with another product called ATA188 in multiple sclerosis.
Thank you. So I appreciate everybody being here. And again, thank you for Arm for hosting us. And so for the first question, and we can just start as a round robin, you know, cell therapy has changed so much. The field has changed so much in the last certainly 5 years, but you know, even before then, I think we could all agree at least somewhat agree that at the beginning we were in the Wild West, so to speak, where maybe not only you were going as a therapeutic developer to the FDA, or even a technology provider in the case of you, Doug, but also an educator. And so I guess my first question to you all is do you miss those days, and what did you learn from them? Maybe we'll start with Kristen on this one.
Sure, thanks, Matt. You know, it's interesting as a working in the allogeneic cell therapy space. I feel like in some ways, you know, we haven't left those days. We're still, you know, really at the brink of advancing these therapies, you know, to approval. Like I said, we're, you know, in the process of preparing filings, but you know, nothing has made it quite across the line yet. So I think we still very much see that education process, the need for a lot of dialogue, a lot of discussion, a lot of education around the need for the products, and then also, you know, what the technology is, what it isn't, what standards are going to be put in place, you know, particularly as health authorities really think about how to evaluate these new products and therapies and their readiness for market.
Thanks. Howard?
Yeah, my perspectives are drawn from a couple of different experiences. The first is really, as I shared with the panel briefly, but when I was involved in the recombinant DNA advisory committee and the early cell and gene therapy products were coming for review, I happened to be a medical reviewer on a couple of the CAR projects that ultimately led to registration. What's become clear to me generally is that there's a lot of knowledge that we generate, and it's important for us to share that in context, not just with the agency, but others. And I think that other constituency are those who actually pay for therapies. Having been a physician, having run a health system, I think the nature of how we pay and what we are paying for and understand the value from the payer's perspective is very important. And I think we often spend a lot of time thinking about the regulatory hurdles, but the most critical one, commercialization, is really thinking about payers. And I think that that's still work in progress for all of us.
I agree. Doug, from the technology side?
So we've been involved in engineering cells for the last 20-some years. And frankly, up until the CD19 drugs were approved, it was really a challenge to get people to think about engineering cells. They were more concerned about raising money, getting the product into the clinic, showing some signs of efficacy and tolerability, and really not taking the risk of an engineered product. And not until the CD19 drugs came on board did we really check 3 boxes. The first box was, yes, there was a therapeutic effect, and an amazing therapeutic effect. Secondly, the regulatory authorities were comfortable with the risk versus the outcomes with these patients. And thirdly, it was thought to be potentially a commercial product. And once we checked those 3 boxes, I think you've seen really a tsunami of capital coming into the marketplace. And over the course of the last 5 or 6 years, I think you've seen a new generation of these more straightforward therapies. Then you've seen allogeneic therapies coming in. But now you're starting to see much more complex therapies arriving, certainly as they relate to solid tumors, where you're doing, multiple engineering at one time. And I think we're, as we were maybe 20 years ago with CD19, I think we're today just at the very, very beginning of understanding the immunology, understanding the therapeutic index of these drugs and how they affect patients.
Right. No, that's a great point. And maybe pulling on the thread a little bit, because you had mentioned, Doug, that, companies in the field are very focused on speed to clinic. And I don't think this is only for cell therapies, but in complex biologics in general. So do you think that will continue? Because what we are seeing, especially in your case, is that companies are beginning to accept and implement more advanced technologies in automation, non-viral transfection, and other technologies, but it still seems as though speed to clinic is the primary concern.
Yeah, I think that could be right, but I also think that the investors who are supporting our companies are much more sophisticated and have a network of people that they can really understand what's going on, and they want to see some early signs of biological impact and efficacy before they make major investments. So, I think the days of just getting a cell into a person and showing its safety and tolerable and being able to raise a lot of money around that, I think is not going to be something that's going to continue. I think investors, again, are going to want to see, they're thinking about this in the long term. They're thinking about what Atara is doing and the years it's taken them to get through.
Right.
It's a major investment, and they want to make sure that they can bind off as much risk as they can and have a drug that is commercializable. And as Howard said, it's eventually going to have to treat people. It's going to have to make a difference, and it's going to have to be commercially viable. So therefore, payers are going to have to step up and feel comfortable with reimbursing these and paying for these drugs.
Right. And so, Kristen, maybe to go to you on this. I mean, going to commercial now, has that been a thought process in terms of ensuring that your processes are suitable for commercial? Are there specific challenges with allogeneic therapies that you might like to highlight about that?
Yeah, so, well, I guess maybe the first thing I would say is, I just loved what you had to say, Howard, about the need for a focus on the value proposition very early on, and really thinking about all of the stakeholders in the healthcare system, beyond just what will be required to get the therapy ultimately approved, which of course involves both the clinical data and of course the CMC data and all of that. So certainly we've been putting all of that together as we approach our filings and preparation of the dossier, but putting together the value proposition, starting to educate not just physicians, but the payers about what is coming in terms of a novel cell therapy. It's important to start that early. It's important not to underestimate the amount of work that you put into it. So for example, one thing that we have found is still, while there's been a lot of learning and a lot more comfort at the institutional level with cell therapy and gene therapies as well, there's still a tendency to think of them as all of the same. So that variety and complexity of different products that Doug was speaking about, when you go and talk to a payer or an institution, which in many cases is also a kind of payer with our products, the tendency will be to just imagine that it's all autologous CAR T, and that there are certain timelines, certain amount of complexity, certain cost expectations that they have around a product like that. And we find that we really need to start very much at the beginning, educating on everything from the disease state itself to what can we do with a therapy that might be able to reach the patient faster in a disease state like post-transplant lymphoproliferative disorder. That's what our lead product is for, where the mortality of patients is very high and they literally have weeks, just a couple of months to live. So really communicating with all of the stakeholders in the healthcare system about what it is that you're trying to do and why it's important and why that's bringing value is something that we've put as much emphasis or time as we've done, building our manufacturing facility or running our trials, but it certainly is a very healthy amount of time and investment.
Right. And so, Howard, I want to maybe go in a slightly different direction and say that, maybe talking to Doug's point earlier, that we're seeing more complex engineering in cells at the moment, as we enter solid tumors. What we're also seeing is that there were a lot of what I would call combinatorial trials happening, where a cell therapy is given in conjunction with another biologic like a mAb or a BiTE or something of that sort. And so I guess my question to you, and we can go around as well, do you think that's something that's going to persist? And if so, do you think it's going to remain a combinatorial approach? Or do you think there will be additional cellular engineering, say using MaxCyte's technology to integrate that ability for the cell to produce a mAb into it itself, as a version 2? Yeah.
Thanks for the question. I think it will depend upon the clinical indication and the mechanism of disease. And so the generalizability of combinatorial therapy is likely to be driven by the need to hit at least 2 different pathways in what is perceived as a pathobiologic network. Cancer is the poster child for this. And because of somatic mutation, it will continue to be, in my estimation. Whether those can be concurrently engineered into a single product, I think is possible, but again, I think it will be clinical indication and context specific. I was going to make a comment, just adding a little bit to Kristen's point. I think what we're in the midst of, coming very much back to where Doug started, is that there's extraordinary progress that's been made to really understand the sophistication of what needs to be done to a cellular product to meet and fully interdict a pathogenic mechanism. And it ties back to your question, Matthew. when we understand mechanism, when we understand how to be demonstrating product against mechanism and have readouts that may even antedate clinical benefit, I think the field is going to shift very quickly to this level of sophistication where it will be multiple edits, and we're going to be pursuing a multiple edited product ourselves. But separately, what will be clear is that there may even be a need to do combination therapy, because you already know enough about that disease to make clear that a single drug product, even if it were multiply edited, is likely to be insufficient to be able to fully interdict the pathogenic mechanism. So to Doug's point, I'll just underscore, we are at the very beginning of what will be a rapid climb to a level of sophistication that we have not seen before. And I would only add one thing to Kristen's comment, is that post-commercialization, the people who actually write the scripts for these products are the other constituency that we need to be mindful of, because if there are other choices to be made with regard to what could have been antecedent standard of care, and these new products may actually be ones that are thought to be based on registrational studies to be even more beneficial, I think that the physician and those who actually are actively looking at what can I do best for my patients, they're going to be the deciders. They're actually going to drive utilization and ultimately, I think, revenue generation.
Right, that's a good point. Maybe going back to a phrase you had said about the foundational knowledge. I mean, certainly it's increased significantly in the past decade or so. Where do you feel we're at there as well? Because there always seems to be a new finding, you know, weekly or monthly in Nature and Science and Cell about new properties that we should be aware of. So where do you think we are, and where do you think we're going?
Is that for me or for one of the other panelists?
Now I'll stick with you for now, and yeah, we'll go around.
Yeah, my sense is that science will always be forging the vector that we who think about product development and harnessing the science to make the best product for whatever the clinical indication. I anticipate that over the next decade, what we think is really sophisticated today is likely to be greatly shifted by new observations, whether it's the next generation of a gene editing protein, one that's more compact or perhaps more efficient with less off-target effects, or perhaps even newer technologies such as those that might eventually supplant technologies for non-viral delivery that are fully tunable and that you can address, and you can get a degree of quote-unquote tropism without using a virus. My overall belief, Matt, is that some things that we will be doing in just 5 years hence are going to be so fundamentally different and better than where the field is now, that when I speak with patients and physician groups, I'd like to remind them that the science is so incredibly powerful and undergirds everything that we do. But we have to decide how to deploy the science in the interest of developing the best therapeutics. And that's what excites me the most.
Thank you. So maybe moving over to you, Doug, to go deeper on this. I mean, obviously gene editing is important in many modalities, whether it's certainly allogeneic and others. And then kind of coming back to this multiple edits foundational knowledge point as well, you know, how does that fit in? And, you know, I think most people believe it will supplant or at least shift from vector to non-viral in the future. So what's your take on that, and how are you preparing for it?
Yeah, so you know, our sense is that there's a greater understanding of the pathology of these diseases, and I think the more tools that we can provide the developers, that they can be more aggressive, more innovative in terms of the approaches. As Howard said, both in the cellular engineering, but also in combination, I think you're going to see some really novel, amazing ideas are going to come up to treat diseases that we never thought one could treat with cells before. I think, though, there's a fundamental issue that we've got to be thinking about, and I want to push Arm on this a bit. You know, one is that we got to talk about the things that don't work. That's really, really important. And I think you can only really do that in an industrial setting where people feel comfortable. And the other thing is we have to make sure that our patent systems are predictable, because if we start to be concerned about our patent systems, we'll start to move away from publishing and keeping things as trade secret. And frankly, I think that's going to really retard the ability for this amazing technology to continue. So I think there's a big policy part of what we had to be thinking about when we move these therapies forward. So it's extremely complex. We've got payers, we got policy, we've got basic science. But I do believe that that folks are looking for ways that they can make these drugs faster, not just get to the clinic faster, but able at the time to treat needs to go down. We can't wait, 6 weeks to treat a patient. We've got to be able to treat these patients in a matter of days for it to be effective. We were seeing that certainly in the allogeneic setting. And we also have to figure out ways of making sure that we can produce these products consistently. It's one thing to produce a product in a single arm study with one location, but quite a different thing to be able to produce that same product in 5 or 6 different locations. You got to make them consistent, and until you can do that, you really don't have a commercializable product. So I think there's going to be a lot of emphasis on kind of industrializing the process, reduce the time to treat, and also being very, very cautious about the cost of these treatments. I don't believe we can continue to develop therapies that are going to cost $500,000 or $1 million a piece. So you've got this kind of Gordian knot. On one hand, you've got complexity that's going up at a pretty phenomenal pace. At the same time, you've got pressures to reduce the time to treat and reduce the cost. And that's going to be a nut that we have to crack. And I think for the field to be successful, we've got to pay attention to that, because those are the kind of things that if we can overcome, I think we're really going to truly see the power of this technology. And I don't think anyone in the field has any belief other than that once we really do harness the power of cells, it's going to really completely revolutionize the way we treat patients and help patients with treating and potentially curing diseases. And we don't usually use that C word, but I think that you're going to start to see that happening with in this industry.
Yeah, I completely agree with you, Doug. And, I might say this is where the evolving toolkit might also help us, sort through some of those, it is quite a Gordian knot, as you said, but, just take the cost and the combination therapy question that you raised, Matt, just a minute ago. Combinations are great, but combinations also cost additional money, and payers are, quite cognizant of that. So, there's also the question of how far can you go with combination therapy in reaching the clinical outcomes and the efficacy that you want, while, maintaining predictability and, some level on the cost that's in line with the value proposition. And just, one program that we've been working on in a partnership with Bayer actually is a CAR T in the solid tumor space. It's directed against mesothelin. This is currently in the clinic, an auto version, and it has a co-stimulatory domain 1XX, which we think will enhance persistent proliferation, but also we've engineered in PD-1 dominant negative receptor. So, I think this is just an example of an approach where elegant or being selective with the right kinds of armoring or edits might help solve the clinical problem, getting the efficacy that we want in a very challenging solid tumor setting. But there may also be the benefit of working on the value side of the equation, where you have put in place already the PD-1 anti-PD-1 functionality through the virtue of that construct.
Right. And I mean, Kristen, just to stay with that, I mean, do you believe that these types of innovations are what we need, or are they going to be coupled with other things such as non-viral gene editing, automation, additional process analytical technologies? What is needed there?
Yeah. No, I think we need, I think we're going to need a lot of it, all of it. It's going to take the village on this to unlock the promise that cell therapy really brings, which, Doug, you spoke to that so eloquently. And cells can do things that macromolecules or small molecules simply can't do. And that's what we're talking about here. But, it takes investment, let's be honest. So, the investment in preparing cell therapies, even to enter the clinic, can be greater than other sorts of therapeutic modalities. I think we do need increasing, automation, control of the process. Certainly, that's been, of heavy interest to regulators, and many cell therapy companies have been, in a lot of dialogue with various health authorities, on that topic. So I think we are going to need, starting with the scientific understanding, as Howard said, applying the right toolkit to create the appropriate cell therapy construct and modality, but then robust, scalable, cost-effective manufacturing, and a rapid logistic and supply chain capability. So I'll tell you, we have really invested a lot just in thinking about logistics. We've been operating in clinical trials sites, 60 sites, 3 continents around the world, and we have been aspiring and meeting the goal of delivering cell therapy product from inventory in 3 days or less for a patient that's waiting for it. So it's building all of these capabilities contemporaneously across the entire value chain.
Great, thanks. Doug, maybe same question to you. What do you think is necessary? I mean, gene editing, automation, additional PAT, regulatory engagement on quicker release, based on short form versus long form testing, open it up and wherever you want to go.
Yeah, I think, the flexibility is going to be important, product consistency. Again, manufacturing, if you manufacture a product in London, it's got to be similar to the one in San Francisco and Tokyo. It's going to be well characterized and, potency. And as we move into situations or areas where you're not necessarily administering these in tertiary care or advanced care hospitals, you're going to really need to be thinking through, how a physician can prescribe these in a way that makes sense to them. Because hopefully for us to be able to treat some of these diseases, we're going to have to, broaden out the patient access for that. And so that's going to be another challenge, that it's really cool science, really cool product. But again, you've got to be able to manufacture consistently, provide it to a patient in a timely manner. And my sense is that we're not anywhere near there. That's the challenge. The good news is we continue to bring in people in the field. And let's face it, maybe 20 years ago, there were very few people who understood what we were doing. I still think we're at the very, very beginning of understanding the science and understanding the impact of cell therapies. But at the same time, we're just beginning to see people coming into this field and recognizing that it's just not a different flavor from biologics manufacturing. It's a whole different animal. And once they realize that, I think we're going to be creating a new generation of scientists, a new generation of folks who understand the intricacies of cell therapy, which I think is, again, going to be moving that knowledge forward. And you're also seeing people moving from company to company, which I think is an amazing part of this industry. And they're bringing with them the knowledge that can help that company not make the same mistakes that maybe were made 2 or 3 times with other companies. So again, these are kind of this has to be a learning environment, because again, this is so brand new, and it's not just big pharma doing it or big biotech, right? It's a lot of small companies, 100-person, 200-person companies, 50-person companies, who really need to create that network to bring that information in and source it with each other so that we can be successful. So I think Kristen said it's a village. It is. We're just going to have to work together. And I think that I hate to again push on ARM a bit. I mean, the ARM's ability to bring these people together at this meeting, for instance, and getting 900 people in the same venue to talk about, the amazing promise of this technology, but also the challenges, and be very frank and transparent about what we all have to do to be successful. And I think again, we're going to get there.
Great, thanks. And Howard, maybe I'll just jump to you because, Doug and I think Kristen as well has mentioned, product profiles. One thing that has been tripping up companies lately in the cell therapy space is potency, which is something that's typically required, from the regulatory agencies. And, how do you believe the potency that we're measuring in cell therapy potentially compares to some of the products that you're developing? And how do you work with regulators to make sure that they have sufficient confidence in the assays you're developing?
Yeah, no, great question. My general sort of overview here is that we need to educate the regulators increasingly more in partnership about identity, purity, and potency.
Right.
I think the issues relating to products that are sourced often from tissues, even though they may be purified, they're probably more heterogeneous than perhaps would be ideal. And so thinking about that from a product development standpoint is something that we've been doing since we've commenced our work. So, the iPSC, which can be differentiated into a mesenchymal stem cell, and IMSC, represents an interesting way to start thinking about your question specifically, where the foundation really is the source of the iPSC, and then characterizing along the differentiated path to the MSC. You can know everything genomically, epigenomically, transcriptomically, proteomically, but the secretome turns out to be really important because these products are intended, once they are infused, to behave in a very regularized, predictable fashion. And I would also add one other thing to this from a clinical trials perspective, is really understanding how in a manner that is specific to your drug product, make certain you understand the biology in situ, not just regarding its safety, tolerability, and perhaps in many indications, longer term clinical readouts, but really looking at other measures that tell you about the in situ biology. So I think you hit the nail on the head. I think that as I've interacted with regulators, I think that this set of questions that you've posed are front and center. And I think it behooves us to, as developers of such drug products, is to make certain that in that partnership mode that I spoke about earlier, we get it right. And we have no claim on hubris, because we probably will sometimes need to come back and revisit it, because we may not get it perfectly. And I think that's the nature of the work ahead. And I think the standards, as they will become more applied by the regulators, whether they be here or elsewhere, is going to compel our industry to be making great decisions at the very beginning. And even though it may seem a little fanciful, thinking about what the product indication and the spec that goes into the product indication when you're actually looking at the label post-BLA. I think that all ties together into work that we have to do, and I think we need to do it in partnership with the agency.
If I could add something, Matt, what Howard's saying, I mean, part of this too is that these are multi-step processes that we're dealing with, and they're biological processes in most cases. And what the other thing we're going to need to really be focused in on is, inline analytics. We've got to be able to manage these products as they're being manufactured. We can't wait to the end of the process. And, frankly, many of the processes that are being developed don't have those sorts of characterization steps built into the process all the way through the process, so that we can actually manufacture and make manipulations within the manufacturing suites if we have to. And that's going to require, again, FDA to be much more open in terms of what they will tolerate in terms of CMC controls, because it's going to CM controls. So a large part of this too, actually, is getting into the actually manufacturing process and making sure that FDA is looking at that from the perspective of cell therapy, not necessarily looking at it as a perspective from small molecule development or from biologics development.
All right. And so maybe to that point, and coupling that with your point earlier around making processes shorter so that we can deliver therapies sooner. And I assume at least in some scenario, you're talking about that in a decentralized fashion. Does potency survive that? Because if you transfect something, it takes 48, 72 hours for it to be expressed, and by that point, you could have already dosed a patient. So, and I think there's another, like a kind of another question here, which is, we haven't linked anything in potency in cell therapy to actual clinical efficacy at this point. So again, I mean, the easy answer is that we will figure it out, which I have confidence in. But if we intend to dose patients in a dialysis method or something within 24 hours, does potency survive as an asset?
The trick to anyone in particular?
Yeah,
Doug, you can take it, and then maybe we can.
I'm happy to take it too. Yeah.
Go ahead, Howard, please.
Yeah, and so
everybody can take it.
Yeah, no, it's probably the most important question. The time to treat is for certain indications, just purely from a clinical perspective, so important, because once that window of opportunity to reverse what could be really a lethal process has closed, you know clinically that you've made either the decision that you'd like to revisit, but you don't get to do it. And so the potency issue is absolutely critical, because you want to make certain, whether it's in an infusion center, in an outpatient center, or maybe even rarely in a inpatient setting, that whatever you're delivering, you know how to measure before infusion something that will then subsequently correlate with whatever clinical outcome you're going to measure. I think that's just the way medicine has to be. And it is incumbent upon us to make certain that we know how to describe that, so that the clinical outcomes, as they stack up in time with more and more patients, irrespective of whatever clinical indication, just hypothetically here, any clinical indication, we need to know that there's a linkage between the potency of the product and the anticipated clinical benefit. And I don't see any way around it. You're right that there will be variability, but I think the thresholds before you may write the order clinically, infuse. They're now corroborated by a nurse and whoever else is in the room if the product is one that is going to require standard infusion. But I don't think we can back away from that. You're right that it will, and again, this is just my sense, and others, I would appreciate your perspective. I think this is sort of common ground for all of us. So if ARM were going to make a priority of this issue, I think there would be a lot of interested parties to sit at the table and figure out with the regulators, how do we get to where we are now to where we think we need to be.
Sure. Kristen, would you like to add anything?
Well, I guess just I think the only thing that I agree with what Howard is saying here. It's also our obligation to be studying this and putting it together in our clinical data and our dossier, right? So I think thoughtful clinical development program that can give confidence in the responses that we're seeing in the data, track back to a potent product, and then that's consistent. That's what we need to do. And I agree that there would be great energy among the ARM community for trying to understand clearly, help regulators think about what's the right guidance to give to us, and that dialogue is in the right spirit. We talked about education and learning at the beginning of this discussion, and I think we're very encouraged. We certainly feel that we're seeing that spirit in discussions that we've been having with health authorities. But I think that spirit needs to continue. It is continuing, and I think ARM, frankly, with things like the series of interactions and workshops that happen every November, it's a great opportunity, and ARM has already really helped frame issues and discussions with, FDA in particular, other health authorities, in the context of those sorts of sessions.
Thank you. Doug, last word.
Yeah, well, biology isn't sequential, right? And so what we're faced with, and even in your example where we want to dose a patient in a day or two, but you're not going to get the results, right? You're going to build some, if you will, surrogate CMC steps. So you're going to be able to have to test something, and I'm not sure the technology's out there. There's new technologies we're going to have to develop. And what's really, I think, fascinating about this industry is the ecosystem between technology developers and therapeutic developers, and that would include CDMOs. I mean, how can we create this again, these partnerships so that we can work together to solve these problems? Because once you solve the problem, you don't have to solve it again if you do it right. And every time you solve that problem, you can advance the industry just another click. And that's what we just have to continue to do. And that requires, excellent business development, corporate development, partnerships, a lot of relationships between technology developers and product developers, and in a way that maybe hasn't been seen before in the industry writ large. But that's again something that I think that, fortunately, with the tools that we have, artificial intelligence, other ways we can move data around, we can do inline analytics, we can do really rapid transfer of information. My sense is we're gonna figure this out, but this is tough stuff. This is sorry to be so pedestrian about it, but I don't think people realize how difficult it is to develop these drugs until you actually start to get involved in understanding the complexity and all the nuances that are involved in actually producing a cell-based drug.
No, I agree. And so maybe sticking with that thread a little bit, how do you think the regulators have done in keeping up with, the pace of the field and innovation and being flexible? Doug, I'll leave that with you as a start.
Well, I think they're learning. I think we're all learning. I think, there's only been a handful of products that have been approved from the cell therapy field, right? And so, I think you're going to see another wave coming out in the next 2 or 3 years. They're going to be more advanced products with, multiple edits. You're going to see some stem cell products that are going to be advancing. And I think every one of those, FDA is busy. FDA's, frankly, they're doing like we're all doing. We're trying to find the right kind of great people to come into our companies. And there's absolutely a shortage of folks that know the field as well as we need them to know it. And that's affecting FDA. So, we also have to figure out ways that we can ensure that FDA has the proper funding. FDA has the ability to bring in the kind of people from industry that can move in and out. Many other agencies in government do that on a routine basis. It helps to build the knowledge base and helps them to become, advancing with the field. And we really have to think this through, because this is different, again, than a biologic or a small molecule. And I just have to give my hat off to the folks at the Cell and Gene Therapy Center and the people who had the wisdom to set this up as a separate unit, because I think that's going to be, really a major requirement for our future, is to ensure that they have the funding, they have the people, and they have the interaction with the sponsors. And I think also being able to have more routine interactions between small companies and true innovators is going to be key for us, the ability to move this forward. Can't wait for, a set meeting 30 days to get an answer. We have to be doing this rapidly and iteratively to be successful. I think they understand that.
Good. Chris, I was going to say, since you're so close, I'm sure you've had multiple interactions, and also being, new modality and allo, how do you feel about this as well?
Yeah, so look, I think, we have the virtue of a breakthrough designation and prime designation in Europe as well. So, we've really enjoyed, a great level of, very collaborative dialogue with health authorities along the way. And I will say that's been, absolutely invaluable, essential. And, I think it was just, as Doug said, that's really what we need as developers of these, you know, new therapies, really pushing, pushing the frontier and trying to bring new options to patients who don't have any and desperately need them as quickly as possible. So, you know, I think we've been, you know, very happy to see, you know, that that level of engagement and dialogue. You know, it's about before, they're very complex. There's a lot to learn. There's a lot of education and understanding, you know, to be reached on both sides. And I would also very much echo what Doug has said about, you know, all of us need to really support the call for, you know, funding and staffing to health authorities and to FDA to, you know, be able to have the level of expertise and, you know, the level of manpower to, you know, so that we work together as a team to advance all of this. And I would call out ARM. I think ARM has been very vocal on this topic and, you know, has sponsored all kinds of activities, including this year's legislative fly-in, you know, that ARM organized with, you know, many of the companies here in attendance today. And, you know, one of the key topics was, you know, talking to all of our Congress people about the need for, you know, increased staffing and funding to FDA and to CBER to provide people with the guidance that's really required here. So I think we're very encouraged with the progress that's been made, and there's a lot more to come. We're all in this for the long road, and as this sector grows and matures, you know, the resourcing to be commensurate with that is going to be very, very important.
And Howard, I don't want to leave you out, of course, in cell therapies, but also in the work you're doing in mRNA. I mean, obviously, you know, mRNA has been around for some time, but, you know, in the past year, in the age of COVID and vaccines, it has certainly come to the forefront in many ways. So, you know, how do you believe that, you know, FDA and others are adjusting to this, these new modalities as well?
Yeah, no, thank you. Just in brief follow-up to Doug and Kristen's great comments, I think first I just would share there's another important pillar here that will enable, assuming that there would be adequate federal resources to staff up and hire great people, whether it be in the FDA or elsewhere. I think the academic institutions who train the next generation should take this on as a problem to solve. There should be master's and doctoral programs focused on the areas that we think are important. I think there should be clinical specialization beyond the typical routine medicine, neurology, pediatrics, where there would be fellowship training, not just for those who have medical degrees, but those who have PhD degrees, so that these will be the people that the agency will want, and certainly our companies would want them as well. And I think there's a symbiosis here, but I think bringing maybe ARM would be the appropriate catalyst, those folks to the table, and think about the enablement of such new didactic programs that actually would lead to a workforce that would be able to step in and do some of the things that Doug started with and which Kristen spoke about. And so back to RNA, I think, you know, we know certainly a lot about the ability to make synthetic RNA. We understand using different types of nucleoside substitutions, but the thing that attracted me and our company is the ability of the cells. It gets back to a cellular problem again, to recognize structure in RNA, and they mount typically a so-called innate immune response. And so the technology that we've in-licensed completely obviates that. And it was the doctoral work of a couple of people that made clear that whether it's the interferon response or whether it's a PKR response, you really need to understand mechanism in order to, as great engineers, engineer into than the synthetic construct that ultimately will be transfected into cells to escape that. So I think the agency is on a big learning curve. The fact that we've rapidly see mRNA go intradermally or intramuscularly, showing at the very least that it's very safe and well tolerated with just a small number of complications associated with it. And in fact, I would consider those to be so minor in terms of the net benefit of us societally and from a community population health perspective. So I think the lid has been blown off this modality, and I think the future with synthetic RNA is so incredible that I couldn't even be able to scale and predict the magnitude of where this is going to be just in a few short years because of what we've witnessed in literally less than 36 months. It's been extraordinary.
Great, thank you. Maybe coming back to a point you made as a follow-on around the next generation in talent. I mean, this field has grown so quickly. I don't think that anybody has anticipated the demand and need for talent at all levels in this industry. So just curious if you have any thoughts or comments on that as well. Maybe, again, just stay with you, Howard, for this as a start.
Yeah, so, historically, I'm a dyed-in-the-wool academic, and so maybe this is sort of coming back to home. I've been fortunate enough to be a dean of a couple of medical schools, so I actually think this is a stimulus that should galvanize a variety of different both patient advocacy groups, industry, regulators, and not-for-profits such as ARM. And so as just a putative mechanism, I think that there should be, at the very least, a roundtable bringing together scientific and medical educators, those who pay for this, so it could include NIH through any of their 21 ICs, and I think having the right host of this, which I believe is going to be an ARM. I think the company should be at the table to articulate need. I think there should also be the regulators at the table because they too have need, although it would appear that it's overlapping with industry. And if there was an outcome, the one that I would find incredible is that one or several institutions would invest their own money with the expectation eventually that there might be training dollars that could come from the federal government. And then offer a variety of opportunities, internships and the like, whether it be in the agency or in companies, and put it on steroids. Because we actually need this, because we're going so fast. Doug started off with this really very, very thoughtful analysis of the pace at which the technology is advancing. We need the next generation to be living it. And the only way I can see doing it is to bring together the right stakeholders to make certain that they're committed to making it happen.
Great, thank you. Kristen, maybe ask you as well, as someone, again, at a company that, is developing a lot of therapeutics, I'm sure requires a lot of folks to be involved, and then, manufacturing them as well. How are you all potentially approaching talent?
We're having discussions about it all the time. So, you always, I think, you sort of judge what are the hot things going on at a company, who you got on speed dial right now. Our chief people officer is on, is on speed dial. So we're being very planful, very mindful about it. And, it's fantastic in that, the company, we're growing rapidly as we approach launch. All of the companies, the sector is growing, so that there is a high demand for talent. And it's very specialized skills and profiles in many instances. So I think from a managerial perspective, you need to be thinking about that very much as well. How engaged is your workforce? What are you doing as a company to drive that engagement? Especially for companies that are smaller, which describes a lot of us, how do you think about career progression and trajectory and growth? What does that mean for people in positions in a wide different variety of functions? So it's something that is on our leadership team agenda just about every meeting, because, you know, we know, and I believe fellow fellows would agree, it starts with the people. It all starts with the people. And if you don't have the right people or the people you need, you know, you're not going to get very far. So investment, you know, dollars, fundraising, all that's important, but it starts with people.
Great. Doug?
Yeah, so I mean, we've been talking a lot about how multifactorial this is. We've got medical people all trying to solve this problem. We've got chemists, we have biologists, we have engineers, we have business people. They all are trained differently. They all think differently. We've got to make sure that we're building cultures in these companies that make people feel comfortable that if they don't know something, it's okay. You say, I don't know it, because the worst thing we can do as an organization is to, you know, kind of fake it till you make it. Some big companies, you can get pretty far by being that way. But I think in small companies, there's no place to hide. And if you're on a fast track as a developer, you can't make that mistake. You've got to be open, because again, we have to realize that we don't know what we don't know about this field. And we've got to create cultures that allow people to feel comfortable doing that. And that's not a typical thing. And we've got to be able to reward these folks. I think that some of the investors get that, because investors are, at the end of the day, they need to make sure that they're supporting companies that they can, you know, draw in and retain the best talent that they possibly can and turn it into a very complex product to treat a patient. And that's probably the most noble cause any of us could ever you know, try to achieve. So that's my thought on it.
Great, thank you. So I think we're at the end here of the panel. So I just want to thank again ARM for putting this together. I want to thank again the panelists for joining me. I hope you all have had a good time. I have. I think it's been a great discussion, and hope everybody enjoys the rest of the meeting. Thank you.
Thank you.
Bye-bye.
Thank you.
Thank you.